Hi, my name's Joe Bellick, and I'm the founding partner of the law offices of Bellick and Fox. We support the mesothelioma community by providing first quality legal representation to mesothelioma patients and their families. We're proud to support the Mesothelioma Applied Research Foundation so they can continue the work that they do every day on behalf of mesothelioma patients and their families.
At MRHFM, we exclusively help mesothelioma patients and their families. It's all we do.
This is why we are proud to sponsor the Mesothelioma Applied Research Foundation and its work to fund mesothelioma research and provide support services and education for patients and their families.
Welcome to this episode of Meso TV. Today we have Dr. Laura Johnson and Dr. Dan Sterman with us. Dr. Johnson is the Chief Scientific Officer at Verismo Therapeutics. She has a background of working at institutions such as Duke, University of Pennsylvania, and GlaxoSmithKline. Dr. Johnson, can you tell us a little bit about your role at Verismo?
Yeah, happy to. Thanks for introducing me, Julie. I, as you mentioned, am the Chief Scientific Officer at a company called Verismo Therapeutics. I also head up all of the R&D activities there. Verismo is a spinoff of the University of Pennsylvania. I actually, as you mentioned, used to be faculty there. And when I was there, one of the great things about it was the collegiality and the way that everyone on the faculty would work together. We're all going for the same cause to try and help eliminate cancer and suffering for patients. And one of the projects that I worked on was actually with a collaborator named Mike Malone. He's a professor in the pathology department there. And he had this great concept for how to make T-cell treatments for cancer better. And he said, we've got this thing, and no matter what you target with it, it just works so well. It gets rid of every cancer you target. You've got to try this in your systems. And I used to specialize in glioblastoma. Now I'm more partial to all tumors. And when I tried it in our system, it was a very challenging model. I mean, this was an intracranial brain tumor glioblastoma model. Nothing could work on it. Nothing could fix it. And I would take the standard treatment we would use that we're going to talk about a little later. It's called CAR T-cell therapy, and put it in with this new modification that Mike had come up with. And it amazed everyone in the lab. It just completely eliminated these untouchable tumors, even very, very well advanced, very large tumors in the animal models. We were all very, very excited about this, of course, although this was during a time when Penn had a large funding agreement with a major pharmaceutical company. And the deal was any inventions that came out of Penn at the time had the first right of refusal to the pharma company. And they were very, very interested in this at first. Then they ended up going in another direction, which ended up being very successful for them. And, it's all good. Anything that leads to good cancer treatments are good. Unfortunately, this project got sort of put on the back burner. So although we wanted to pursue it, the company had still owned the rights to it. And so University of Penn went back to them and negotiated for, I think it actually took them 6 years to come to the point where they had an agreement that Penn could revert the rights and start a company based off of this technology. It was exciting. It had people wanting to invest in it. It had backing. It had a fantastic CEO. Our CEO, Brian King, was involved in it. And Mike Malone one day gave me a call. I was at GlaxoSmithKline at this time, running their cell therapy unit. And he said, "Hey, remember that amazing project that we worked on together?" And I said, "Yeah, whatever happened to that? Is it in trials? What has it done for patients?"
Yeah.
Well, it turns out we're turning it into a company. And how would you like to come and run the science for it? And so I did a little bit of research, a little bit of background, make sure the
Sure.
people were solid, and they absolutely were. And that was almost exactly 3 years ago, July of 2021. We opened our doors, 4 of us at Verismo. And to date, now we have 2 INDs, 2 clinical trials, a healthy pipeline, and we're incredibly excited to see what this can do in patients.
That is exciting. And we also have Dr. Dan Sterman here with us. I'm very excited. Dr. Sterman is a interventional pulmonologist at New York University. And could you tell us a little about yourself and what you do in your sort of collaborative role with Verismo?
Certainly. Let me take you a little bit back. My history overlaps a bit with Laura. In fact, overlaps a bit with Mike Malone, as Mike was my intern when I was an attending in the medical intensive care unit at the University of Pennsylvania. And I tried to convince Mike to become an interventional pulmonologist like myself. And I failed miserably, and he became a brilliant scientist and successful medical entrepreneur. And I take great pride in trying to recruit him way back when. But my history at Penn is that as a pulmonary critical care fellow, I entered a laboratory called the Thoracic Oncology Research Laboratory, co-led by one of my primary mentor, Stephen L. Belta, and Dr. Larry Kaiser, who was the head of thoracic surgery and later the chairman of surgery. And this laboratory was dedicated towards taking novel technologies that were being developed in the Institute for Human Gene Therapy at Penn and applying these to refractory diseases. And at that time, in the early 1990s, the most refractory malignancy that we can imagine, and with all due respect to glioblastoma, was malignant pleural mesothelioma. There were no approved treatments for mesothelioma in the early 1990s prior to pemetrexed being approved. And we thought that in the early days of human gene therapy, that this was an excellent target. And so I spent my years in the laboratory developing novel therapies, first in murine models and then in human clinical trials. And then when I came on faculty several years later, I was the co-founder and co-director of the Penn Mesothelioma and Pleural Diseases Program, which is one of the world's leading programs, both in clinical care and clinical research. And around the same time, I was elected president of the International Mesothelioma Interest Group and has been a member of the board of directors and the scientific advisory board for the Meso Foundation. And so the majority of my career has been dedicated toward developing novel therapies for mesothelioma and lung cancer. And during this time, in the late 1990s and early 2000s, one of my other mentors, besides Dr. Albelda, in the early days of CAR T-cell therapy was Carl June at the University of Pennsylvania. And myself and our team in the mesothelioma program at Penn were very much involved in the early days of developing CAR T-cell therapies for mesothelioma. One of my mentees, Dr. Andrew Haas, still leads many of the clinical trials at Penn, investigating the role of CAR T therapies. And we're very hopeful that CAR T therapies, including the Syncure CAR that we're going to be hearing about today, will allow us to mimic the success that CAR T-cell therapies have seen in liquid tumors, that we'll see them in the future in solid tumors. And in some interesting conglomerations, my area of research in oncolytic virus therapy and CAR T-cell therapies may overlap. And if we have time, I can talk about the potential overlaps in the future.
Thank you for both of you for those really impressive introductions. So as we know, this is Meso TV, and you gave us a little taste of that. And we talk about mesothelioma. Verismo has a trial, STAR 101, that's now open. Can we hear a bit about this trial, how long it's been open, and where, Dr. Sterman?
Sure. So let me just go over the title of this trial first, so everyone is aware. It's a long title, but I think it's important because it's It's a trial that's targeting more than just mesothelioma.
Yes.
So it's called the phase 1 study of SYNCURE 110, which are autologous T cells transduced with mesothelin cure CAR in subjects with mesothelin-expressing advanced ovarian cancer, cholangiocarcinoma, or mesothelioma. And the mesothelioma can be either pleural or peritoneal of the epithelial subtype. And the purpose, this is a phase 1 study. So for those in the audience who are not in science or in medicine, phase 1 trials are primarily to take a novel therapeutic agent and to find often the correct dosage, to see what the safety of it is, and to look for early signals of efficacy. Again, most phase 1 trials are not designed to say that a novel therapeutic is effective or is not effective, but we hope to see some signal not only of safety and dose finding, but that it may be efficacious in the tumors that it's targeting. And so the goal here is the same in this trial: safety, dose finding, and evaluate the adverse events and hopefully signals of efficacy. And this trial started recruiting patients in March of 2023. And right now there are 4 sites across the United States where the trial is open, and soon we'll be dosing patients. University of Pennsylvania was the first site open, not surprising considering Verismo's relationship to the University of Pennsylvania.
Yes.
MD Anderson, the second site, and then more recently University of Wisconsin, and then the University of Kansas. So really the goal is to have a broad base of cancer centers, experience in a variety of tumors, be involved in the clinical trial.
Okay. I think we should probably back up a little and have Dr. Johnson explain exactly what is CAR T. We hear a lot of abbreviations like stem cell transplants, T cell trials. These are some of the terms I hear patients come to me with. And can you explain exactly what is CAR T and how does it work?
Thanks, Julie. Yeah, absolutely happy to. CAR T cell therapy, we're in a really exciting time right now. Things have come together over many years and many decades recently. And some of the people Dr. Sterman mentioned, Carl June, is really sort of at the pinnacle of CAR T therapy, finding, understanding, translating into patients. Along with another previous mentor of mine, Dr. Rosenberg, when I did my postdoctoral fellowship at the National Cancer Institute. He also was one of the first pioneers with that, and he has actually gone on to work more with the cells that are already existing in patients' tumors. And they recently had that approved as the first solid tumor T-cell therapy for patients globally. So that was a very, very exciting moment that just happened this year, actually. So I'm going to tell you a little bit about CAR T therapy, which is an aspect of what we call adoptive cell therapy. And the reason it's called adoptive cell therapy is really it takes a patient's T cells and transfers them, sort of adoptively transfers them from the lab back into the patient. And the way that we do this, particularly in this trial, STAR 101, we have something that we call a cure CAR T-cell therapy. So it's an aspect of CAR therapy. This study involves research of, as Dr. Sterman mentioned, it's an investigational treatment called SYNCURE 110. So CAR T-cell therapy is really a type of treatment in which a patient's own T cells, and T cells are the white blood cells that are part of your circulating immune system. You have them just in your regular blood all the time, every day, all day. So the way that we get these is we take just a regular blood sample. We do something called an apheresis, and it allows us to enrich those white blood cells, the T cells, out of a patient's blood. We then are able to modify those in a laboratory, and the modification really just helps retarget these cells so that now they're able to recognize and locate cancer as being bad. The problem with cancer is often it looks too much like you. It looks a little bit too good. And so your T cells don't generally recognize it as being dangerous. So what this allows us to do is to tell the T cells exactly what on the cancer is different, so that when they encounter it and find it, they can actually activate and destroy those cancer cells directly. So these immune cells are then, as I mentioned, modified in the laboratory to be able to recognize cancer cells. We're then able, again, in the laboratory to expand them and grow them into very, very large numbers. Then we infuse them back into the patient. And by infuse, it really is very simple. It's just in an intravenous drip from a bag, and it takes maybe 10 minutes. Dan can back me up. He would know more about how long this takes at the bedside. I'm the lab half. And these T cells are called chimeric antigen receptor T cells. And I'll tell you a little
Hence the name CAR T. Yeah.
little bit about why they're chimeric. In particular, this product, SynCure 110, recognizes a protein that's called mesothelin. Mesothelin is widely and frequently expressed on solid tumors, in particular cholangiocarcinoma, ovarian cancer, and mesothelioma.
Yep.
So it's when these T cells encounter this mesothelin protein that they're able to recognize, hey, this is a tumor, and they activate and hopefully eliminate the tumor.
So
Yeah,
it's
a
kind
couple
of like
things
a
real quickly, Julie,
heat-seeking
about-
missile, right?
Julie, can I add a couple of things to what Laura just said, if I may? Because it's
Sure, of
important for
course.
those watching Meso TV who might be interested in this clinical trial to understand that there are different subtypes of mesothelioma. So when
Yes.
you're diagnosed with mesothelioma and you have a biopsy performed, that biopsy is sent to a pathologist. And the pathologist not only makes the diagnosis of mesothelioma, but informs the clinician and the patient about the histologic subtype. And there are different levels of mesothelin expression on mesotheliomas depending upon the histologic subtype. So the most common histologic subtype of mesothelioma is epithelial or epithelioid. And epithelial mesotheliomas almost always express very high levels of mesothelin on their surface. You can essentially assume that an epithelial mesothelioma, either of the pleura or the peritoneal cavity, will express mesothelin at high levels.
Yes.
The sarcomatoid variant, which is about 10% of all mesotheliomas, does not express mesothelin at very high levels, and then about 20% of mesotheliomas are so-called biphasic,
Yes.
and they have features of both sarcomatoid and epithelial mesothelioma, and their levels of mesothelin expression are obviously less. They're epithelial predominant and sarcomatoid predominant biphasics. And in this trial, we're primarily including patients with epithelioid who have high levels of mesothelin expression de novo on their cell surface. So I think it's really important as patients and families are watching this that not all patients, but the majority of patients who are diagnosed with mesothelioma are potentially candidates for the study.
So, Dr. Johnson, could you tell us a little about the patient journey, or what a patient would go through if they were to consent to this trial and the process?
Absolutely. Happy to. Thanks, Julie. And we also have a slide depicting sort of the patient
Yes.
journey, as you just mentioned right here. And really what happens is the patient would come in. They would see their referring physician. They would let them know about the trial. First thing, of course, is to sign an informed consent. It's where you learn a little bit more in details. The patient then undergoes a series of screening tests. A lot of it, the doctor will already know. So this will just make sure that they match the inclusion criteria and don't have anything on the exclusion criteria. That they have a scan done to show that they have measurable disease. Usually this takes in the sort of days to maybe a couple of weeks to have done. At that point, once they are enrolled on the trial, they will then schedule an apheresis, which is, again, like a blood draw, but it takes usually a couple of hours. And this way they really enrich these circulating T cells from your blood into the blood donation that you're giving. Then that is shipped off to the cell manufacturing site, where they start the process of growing the cells in the lab. They change them so they can now target them to recognize the cancer. Then they expand them. And that takes a little while. They then undergo extensive safety testing, soup to nuts, to make sure that everything is absolutely okay before they're ready to go back into the patient. I might mention that because this can be a lengthy process, it is okay for the patient to go on some form of bridging therapy, bridging chemo, if they think that this is the way to go with their treating physician, that's perfectly fine according to this trial, as long as they finish in time for the sort of 6-week production. Then they come in about a week before they're going to get their cell infusion. They get their baseline markers done. So they'll get their scans done, they'll get their blood tests done. We require a tumor biopsy. That way we can know exactly what their tumor looked like, what was going on at the time of their treatment. This really helps us to understand later, if this patient has a biologic response or doesn't have a biologic response, what were the contributing factors? How can we utilize these to make the treatment better in the next iteration, if that's the case? Or perhaps a certain patient had a marker that we can evaluate that says, oh, these patients tend to do very well. We can look for this when we're enrolling our patients. So these are all very important to the researchers as well. So that's about a week beforehand. Then they start what's called a lymphodepleting chemotherapy. Now, what this is it's a chemotherapy. It's 4 days of a combination of something called cyclophosphamide and fludarabine. These are actually typically used in heme malignancies or blood cancers for patients, because what they do is they eliminate all of your circulating blood cells, your circulating white blood cells, the T cells, not your red blood cells, you're fine, and not your stem cells, so they come back. But when you eliminate the circulating T cells, what happens with T cells, and it's really quite amazing, as a biologist, I'm constantly surprised that if we looked at your blood, if we looked at my blood, if we looked at Dr. Sterman's blood, just about anyone, we'll have a certain number. Just say it's 1,000 cells per microliter. We'll all have that, give or take a few, one direction or another. We don't have more than that, and we don't really have less than that. So what that means is, if you remove the cells in the body, when you put fresh cells in, and the fresh cells that we put in on day 0 are our tumor reactive T cells that we've made. Now, those T cells will circulate in the patient, and they will expand and expand and expand until they reach about, you guessed it, 1,000 per microliter. So it's sort of a way we do an expansion outside of the lab, and then the human body does it themselves if we provide this pre-treatment lymphodepletion. So it really sets the stage for receiving it and making all of their cells, these cancer cells, cancer-treating cells. So that happens at day 0. This particular trial, it's written so that they can have about a 48 to 72 hour inpatient. So they can leave it as an inpatient just for safety. It's a first in human phase 1 trial. Don't expect any problems, but this way we ensure it, that we'll see what's going on with the patient at the right time. And after they're discharged on day 1, day 2 afterwards, they'll undergo a series of safety assessments when they come in, every few days for the first week, then weekly for the first month, then each month until the 3rd month, then month 6, month 9, and month 12. And they'll have scans throughout that time so we can monitor not just how their blood is doing, how the cells are doing in their blood, but also take a look at how the tumor is responding or not to this. And then after 12 months, they would be invited to enroll in what we call a long-term follow-up trial. And that's something that's mandated by the FDA. They just want anyone who receives any kind of gene therapy to be followed for at least 15 years. So that's another trial. So that is the patient journey.
I have a question for you, Dr. Sterman. CAR T-cell, I know, has been very effective, and it's even been FDA approved in certain blood cancers. And can you speak to the success in that, and why is it that's been so successful in blood cancers?
Yeah, I don't know that everyone knows why CAR T-cell therapy has been so much more successful in liquid tumors, so-called blood cancers, than it has been in solid tumors. That's part of what we're trying to elucidate with many different studies, including at Verismo. But I was privileged to have been at the University of Pennsylvania in the early days of CAR T-cell therapy. targeting molecules such as CD19 on the surface of certain cells, like B cells in the peripheral blood. And these have proved absolutely transformative in the treatment of leukemias and some lymphomas, and were the first approvals of CAR T-cell therapies for treatment of malignancy, and have really led to, in many cases, long-term remissions and even cures of what had previously been incurable malignancies. There are a lot of hypotheses. If you can imagine, if you're infusing these modified T cells, these modified cells that are essentially targeted directly against cancer cells, and they're infused to the bloodstream, they're contacting all the time these tumor cells that are also existing in the bloodstream. And there aren't a lot of other cells surrounding the cancer cells that would prevent the CAR T cells from attacking the tumor cells. It's a much different scenario in mesothelioma or in cholangiocarcinoma or in ovarian carcinoma, where the CAR T cells have to get into the environment of the tumor, the interior of the tumor, where there are a lot of barriers, just physical barriers as well as cellular and other chemical barriers that may try to prevent the CAR T cells from attacking the tumor cells and killing them. So we hope that we can be as transformative in solid tumors in the next decade as liquid tumors have been in the last 10 years. And as Laura said, these tumor-infiltrating lymphocyte therapies for melanoma that were recently approved, we hope will lead to other cell therapies that will be approved in mesothelioma and other solid tumors in the future.
Yes, and that led to exactly into my next question for Dr. Johnson, which was why the success has not translated into solid tumors like mesothelioma or ovarian cancer. If you could speak a little bit to what Dr. Sterman started on, which was, I think, sometimes called the tumor microenvironment, or what is it that makes it harder to have that same success in solid tumors?
Absolutely, yeah. The CAR T cells, and I think that you may have a diagram that you can share with the audience here.
Yes.
Titled we look at it as a paradigm shift in CAR T cell therapy. Mm-hmm. So what CAR T cell therapy really is, and I mentioned this earlier, I preluded to this, that chimeric antigen receptor therapy. So the chimera, if you are aware of this, in Greco-Roman mythology, it was actually a mythological beast, a monster that didn't exist in nature. And it had the head of a lion and the body of a goat with the tail of a snake. And it's it sounds very funny that this is something that you would name this kind of a cellular medicine after, but when you see what it is, it's actually extremely appropriate. And I have a slide here with a picture, and it shows on the far left, this is sort of how CAR T started. We call it CAR 101 or 1.1. And it was actually amazing, the foresight that some of these pioneers had. A researcher named Zelig Eshar, in particular, was someone who came up with this idea 25 years ago, almost 30 years ago now, that if you could take a receptor from a B cell, and what makes B cells special is B cells are what makes your antibodies. And antibodies are special in that you can target those to bind literally any protein that you can think of. So he said, hey, if we take the binder from a B cell and we recombine that with the body of a T cell so that it activates like a T cell and destroys its targets like a T cell, because T cells normally target things like viruses and pathogens and infected cells and things like that to eliminate them, then we could put that back in a T cell and redirect it to any protein we wanted to. And then when it encountered it, it would eliminate it. Great idea. Unfortunately, it took almost 20 years to get to the point where technology caught up with the concept and they were able to do that. Yes. And I show sort of a diagram here. If you take a look underneath a series of squares, there are sort of beads on a string looking molecules.
Right.
There's a little white zipper. That zipper actually represents the outer membrane of a T cell. So if you think a T cell is sort of a global ball, that would be the membrane dividing the inside of it from the outside. The orange represents this antibody, the binder, and the blue portion is on the inside of the cell, and this represents the T cell activation signal. So the idea was if you could do this, then they would put these back into patients and have them recognize the tumors and eliminate them. So as I mentioned, 20 years later, they were finally able to do this in a way that they could try it in a clinical trial. And some of these very, very early clinical trials targeted antigens called CA9. And this was supposed to be something for sort of biliary carcinomas. And when they put these T cells in patients, they didn't really see anything right away. And it was sort of a good news, bad news scenario. They didn't see anything right away, except the patients reported some toxicities, some problems, some discomfort. But those rapidly went away. And when they looked for the cells in the patient's body, they couldn't find them after they'd given them. And it turned out that, yes, CA9 was being found, it was being eliminated, but it also was expressed on normal cells in the biliary sort of ducts coming out of the liver. And so it was finding those and eliminating those. The lucky part is that these initial, we call them Gen 1 T cells, they didn't last very long. So they were sort of a one-hit wonder. They would find a target, they would hit it, but then they would self-eliminate. And it turns out I'm an immunologist by training. And, if you spoke to an immunologist, they'd say, oh, well, that's because a T cell's first job is not to harm yourself. Do no harm. And once you get past the do no harm, then go and eliminate things that are dangerous or bad for you, like viruses and pathogens and things like that.
Right.
And the way that a T cell knows good from bad, it's called a two-signal system. So if it sees a particular protein, then it knows to eliminate it, but only if that protein comes along with something like a danger signal or a foreign signal that the T cell recognizes as not self, then it gets that feedback loop that says, I recognize this and I recognize that it's bad. So I'm going to go ahead and attack it and continue doing so.
Right.
So people, researchers thought about this and went, you know what? Well, we can take this. And they made what we call Gen 2 CAR T cells. So they've still got the binder on the outside that can recognize a protein. They've got the internal T cell activation, but now they add something called co-stimulation. And co-stimulation is the equivalent of one of these bad signals or danger signals. So now you have a binder with a co-stimulation and an activator, and believe it or not, they worked. And they didn't just work, they worked really well. And it was a bit serendipitous, I think, that one of the very first targets that researchers looked at happened to be one on these blood tumors that Dr. Sterman was talking about. And so when they did use these in patients, I think no one was more surprised than the treating physicians and researchers themselves that the tumors just went away. They absolutely were eliminated. They were eliminated so fast, they actually had to figure out how to take care of the patients because their tumors were disappearing so quickly in them, exploding. But they've got that pretty much down pat now. And those are the CAR T therapies that are approved by the FDA and actually globally today for the different blood cancers. And they seem to work very well. So that's what we call CAR 1.2 or second generation CAR Ts. Then, as Dr. Sterman mentioned, people tried applying them to cancer, solid cancers, and the same thing didn't happen. And it was really frustrating because, one of those researchers that was doing it at the bench, you would target these tumors in a dish, you would do the exact same thing as for the blood cancers, and they would work on the bench top.
Right.
But then when they would try and put these into patients with these tumors, and Dr. Haas, Dr. Albelda, these were some of the first researchers to actually attempt this in patients with mesothelioma, with ovarian cancer, with pancreatic cancer.
Yes.
They found that while it would initially go to the tumor, they could see a little bit of shrinkage even on the scans of that patient, but very rapidly thereafter, the tumors would just grow out, the T cells would disappear, and nothing would happen. And that was very frustrating because normally when you target something in a dish, it's going to work in a patient or in an animal model, and that wasn't happening. So researchers then said, well, okay, let's go back to basics. If we have that binder and that one danger signal that's good, maybe two danger signals is better. And so that ended up being what we now call CAR 1.3 or third generation T cells, our T cells. That's shown here on the right. And actually, while they worked again in the dish, they didn't really do any better. And they started to reach the limitations of what we were able to do with molecular medicine, with being able to transfer these genes into human cells. They were sort of reaching their size limit. So they had some drawbacks. So then they moved on to something called fourth generation CAR T cells, which is really where the field has been for about the last 10 years, 7 to 10 years, these fourth generation or CAR 1.4. And all that is we've gone back to the initial ones that worked really well, these second, but now you can use these as a delivery system where you can have them target a tumor, an antigen, whatever you want, and they can deliver a little something as a payload. Maybe they can deliver some chemical that helps the T cell to survive, or something else to stimulate the area to be able to get into the tumor better, as Dr. Sterman mentioned. Great idea, great concept. I really hope they work. It's been some time now, and we haven't really seen any positive clinical results, so the jury's out. But this is sort of where the research that Mike Malone and Carl June and we were doing at the University of Pennsylvania comes in. And this is
where Verismo
came from, really, literally. And, Mike is, he's a very smart guy. And he sort of stepped back one day and he said, why is this working in blood cancers and not in solid tumors? It doesn't make sense. Why is this happening? And if you go back to basics and you just go through sort of the logic of it and the common sense, it's like, well, a solid tumor is solid. It's packed with all of these tumor cells and other cells that we know are there that work to do different things in the tumor at the same time. And so something with that must be the difference. And if you look at the CAR T itself, okay, why is that not working? But regular T cells and B cells and other cells do work long-term for the life of a patient to fight a virus, to fight a pathogen, something like that. What's the difference? And if you look closely at the CAR T cells, now it's in the name, it's literally in the name. You have taken the head of a B cell, put it through a Frankenstein neck into a T cell, and put it on the body of a goat with a snake's tail. And even though here these are depicted kind of as beads on a string, they're not separate proteins that are closely tied together. They're actually literally the top half of one protein, Frankenstein stitched to the neck of another protein, Frankenstein stitched to the tail of a third. And that figuration doesn't exist in normal cells in nature. What doesn't exist is this outside of the cell, inside of the cell being on one protein. And I'm going to talk with my hands a little bit here.
Okay.
What this does is, as the T cell moves around, and it's really quite exciting if you can ever see them through a microscope, they literally crawl right along your cells and in different things. And what that does is it allows the receptor, the outside binder, to sort of sample the environment everywhere it goes to say, is this good or is this bad? Which is normal. What's not normal is that it's physically attached to that internal activation. And that internal activation, just that movement, that stimulus, is enough to give it a low activation signal So
Mhm.
what we have are these CAR T cells are constantly active at a low level. They don't seem to be doing anything that causes toxicity in patients. It doesn't seem to cause any damage.
Mhm.
But what it does do is it doesn't allow that actual T cell to ever stop signaling, rest, and recover. And just
Yeah.
like, every creature in nature, including me, for sure, 8 hours a night, please.
Yep. Yep.
If you can't rest and recover, you can't go out and fight another day. And T cells
Yes.
are no different. And so in patients that are receiving these kind of T cells for solid tumor therapies, they're finding that the T cells are becoming very exhausted and literally just giving up the ghost. They stop working entirely. So Mike went back to nature and he said, well, okay, why do normal T cells last forever and keep working and these don't? He said, well, okay, the binder is on the outside of every type of cell that exists, B cells, T cells, everything. And the signal is on the inside and it's completely separated. They don't normally touch, they don't talk to each other
Yeah.
unless and until that target on the outside binds that binder on the outside, which then calls the activation protein to come close to it and then signals as one protein to activate the cell. And you can sort of see in the diagram here,
Yeah.
it's the orange on the outside is the receptor and the blue is the signal.
Sure.
So the reason we call these KIR CAR is this is the actual existing natural signal that is called a killer immunoglobulin-like receptor. Big word, KIR, KIR CARs. And NK cells, which are another type of immune cell in your body that fights cancer, T cells in your body also can have this type of signaling naturally occurring. So really it's going back to nature to try and come and solve the problem. And that's where we get the KIR CAR from. And so really in sort of the next slide, you can see it's designed that when it's at rest, these proteins exist, but they exist separately. And the T cell can be completely rested down, doing its surveillance, testing its audience, testing the background for what happens. Only when it finds its actual engaged protein does it then activate. So this independent binding and this independent signaling chain, they separate the binding from activation. The chains come together and form a single receptor complex when they're bound to this target being a tumor, in this case, mesothelin on mesothelioma tumors. The KIR CAR then provides prolonged T cell activation, even in challenging solid tumor microenvironments. And this tumor environment that Dan's alluding to, there are all sorts of other immune cells there that the tumor hijacks, and their job is to try to turn the T cells off. And they have evolved to do that very, very well, unfortunately. The tumors take up all of the nutrients and the sort of micronutrients in the environment and sucks them away from the T cell to try and starve them at the same time. So now they're starved, they're exhausted. It's a pretty tough case, and they're getting attacked by an army of other cells influencing the tumor. So adding that extra Frankensteinian protein, the chimera, is really sort of the last straw. It works, and it works until the cell just gives up the ghost. So, the idea is that these KIR CARs have shown enhanced surface stability. They've shown enhanced signaling and receptor expression in both the activated and the resting state. That, touch wood, at least in our longer term preclinical models, has shown that it allows them to keep working, to keep fighting the tumor until every last cell is gone. And this is our goal for this to happen in patients.
Julie, if I may add just a couple of comments and
Sure.
to Laura's brilliant exposition of.
That was a wonderful
I learned so much from
explanation.
her just listening to her, even as someone who's not a layperson. Two things. One is the KIR CAR construct that Laura described is one way of dealing with a very common problem in cancer immunotherapy. So those who are watching the show know that there's been a transformation in how cancer is treated, that there are a number of drugs out there CAR T cells are just one type of immunotherapy that are being implemented, studied. Many of these immunotherapies have been approved, including in mesothelioma. And a big problem is this idea that the T cells, which are the workhorse of any immunotherapy to try to target and attack and control tumor cells, that these cells become exhausted. And this is the limitation of almost any immunotherapy, including the drugs that people see advertised on television. The limitations, especially in solid tumors, for the efficacy of these drugs is that the T cells become exhausted. And so in many areas of immunotherapy, researchers like Laura are investigating ways to try to avoid or overcome this exhaustion to allow for better tumor cell killing by the T cells. And so in CAR T cell therapy, this new construct is one way of avoiding the exhaustion. The second thing I wanted to add is just to talk about the differences between animal models of mesothelioma and humans. And I think this is important about when people who are lay persons or patients and families are reading about preclinical experiments and then try to figure out why they're not working as well in humans with the same disease.
Yes.
Having worked in animal models, mouse models of mesothelioma for a lot of my career,